Thanks, Greg, and thanks for inviting me. Is the microphone working okay? Yep. Good. All right. Excellent. So it seems like it's been a great summer school so far on theoretical biophysics. There is a bit of a question, though, as to why on earth I'm here. Right. So you look at everybody else that's gone through, and as you've seen so far in the talk so far, and people have largely been able to write down things in terms of simple models and other sorts of descriptions. Whereas if you think about, like, what's the prototypical example of physicist hubris of what we can't do, what is it? A spherical cow, right? And so precisely what I'm going to do here is try and figure out what, if not a spherical cow, what type of cow? Ahmed. I'm not even going to go there. I'm on YouTube. So the idea is that, like, we're trying to go and think about animal behavior, this really super complicated thing where, from the standpoint of a theoretical physicist, and understanding this is going to be in a little bit different way than other sorts of things you've seen. Like you see this beautiful work that Andrew presented about cell sensing, where you can write down a reasonably simple framework and then go and solve a bunch of equations and smile, and then those equations are then tested in an experiment. You can smile even more. Or you see other things, even in the case of the stuff where Michael was talking about it, and you could write down these equations, and the equations might be pretty hard to solve, but at least you could write down the equations. I don't even know what the equations are. You don't even know what the observables are here. And that's what we're going to try and sort of discuss for a minute. So sort of as a prologue, and because we're just coming on the heels of a machine learning talk, it probably would have been slightly more appropriate to go talk about cats, but we'll talk about dogs. So prologue one. So let's say we've got a bunch of dogs that we're observing in the wild. And let's say that we have really good experimentalist friends that can measure whatever we want. As good theorists, we should be able to say, you should go and measure that thing and tell us about this. What are the quantities from and you can start blurting some out. What are some of the quantities you might want to measure about just a bunch of dogs roaming around? Separation. Yeah, so you might want to say something about, I'm going to expand out, and let's just say positions. Right, so where are they? Yeah?
Cluster sizes?
Sorry?
Cluster sizes.
Yeah, so so I guess this is similar to also what Andrew said, but say relative position. Relative position and clustering. Like, how many of them are sort of nearby each other? All right, what else? Yeah, sort of something about well, in some ways that's partially about positions, right? So, but there's maybe, let's expand this to sort of position relative to the environment. So maybe it's not just the range. Maybe there's some pole in the middle that you're wanting to see where they are. Maybe there's a den where they slept at night. Okay, what else could we measure? Yeah, you could measure, so broader, we'll say kinematics. So we could measure velocity, angular acceleration, jerk, whatever you wanted to measure, right? What else?
Activity, like are they going out to another animal?
Right, so relationship to non-dogs, or wolves in this case, but we'll call them dogs because it sounds cuter. They're basically the same. What else? This is so all of these things require one type of measurement, right? You're measuring the center of mass of where that animal is located. What else could we measure?
Posture.
Sorry?
Posture.
The posture, right? So we could measure the posture, so that the location of all of their limbs and where their snout is and all sorts of much more detailed information. What else? Yeah, Liam.
The complete sensory input of
Yeah, right. Yeah, we have really good colleagues. Right. So we could measure, yeah, sensory input. I mean, we could also do things like, let's say we put backpacks on them, right? We could measure like heart rate, other physiological parameters. We could measure all sorts of, yeah, Russell. Yes, right. So, yeah, so maybe we'll say sensory input / reactions. Yeah, so I would sort of put that in sort of like relative positioning / clustering, right? Like how are they talking to each other? Which ones are they nearby? But the point is, these are a bunch of measurements. Where's the theory? Right. What's the question we're going to ask about? I mean, we can ask questions like, okay, how much are they clustering together? We could write down a V-Sheck model to say these sorts of things. But when we're thinking about behavior, and what we're going to talk today, and what I'm going to focus more on, is less on the center of mass stuff, and more on sort of stuff, sort of on type, sort of like this scale here, right? Thinking about how do we actually measure behavior from an animal, and then sort of scale that outwards. So Lisa talked a lot about V-Sheck model stuff already, and we might come back around to that on the last lecture, which may or may not be about social behavior, depending on whether I get it written before that or not. But the idea is that we essentially are going to try and zoom in and look at sort of how this posture and how kinematic changes, how we can measure those things. But just from measuring a kinematic, right? From measuring kinematics, how could you tell if there's a social interaction? Right? This is not trivial, right? You could make some assumption like, oh, if animal comes within some area, oh, that's therefore a social interaction, but we don't actually know these animals. We can anthropomorphize, we can look at them, we can make our best guesses. We don't know what's actually socially important for the animal just by looking at it. So we need to try and build up sort of very detailed ideas and very detailed models to try and figure these sorts of things out. Right. So that's sort of measuring dogs in the wild. Right. So that's prologue 1. Prologue 2 and 3 will be much shorter. Prologue 2 is Pavlov and his dogs. Right. This is a very famous psychology experiment. For those of you who haven't seen it before, you put a dog up and you ring a bell each time you feed it, so then it starts to salivate each time it hears the bell. And what they can measure here, instead of doing all these things, is they've designed an experiment so that each time, let's say the bell may be ringing, the bell may not be ringing, but you can actually design sort of a fancy setup so you can actually have a time series for the amount of saliva that's coming out. And you've made that measurement because you know something about the specific question you're interested in. You've observed some salient behavior, and you've encoded that measurement into your experiment in and of itself. So now the next time the dog hears the bell, it'll start salivating just that, and you can see the little peak, whereas originally it might have just started salivating when it smelled the food. And so we saw those things. But now let's see, we have an even more detailed interaction. And this involves, to me at least, much less abstracted animals. These are my dog and cat. How do you put numbers on this? How do you quantify this interaction? Right? How do you actually try and think about what's happened between those two? Like, you look at it and you can tell that there's some reason why these animals have done or acting the way they are, but pulling it out directly from data and writing down a theory where we don't have to put our own sort of assumptions as to what that might look like is rather difficult. And we're going to get into that a lot. But I want to sort of set the stage. So let's think a bit about, let's go down. So what I want to ask, I'm going to ask you guys more questions because it's after lunch and I know there's probably a food coma going on. So why is this hard? Okay. So what actually is it about animal behavior that makes it in some ways more difficult to write down theories than, say, looking at evolution or looking at cell sensing. Not to say that those things aren't difficult to actually solve the equations, but I'm even just talking about the setup. Why is animal behavior so tricky? Yeah.
Dimension is high.
Mm-hmm.
Also, maybe individual
Sorry,
individual animals do different behavior.
right, so we have dimension is often very high, although we'll see it doesn't necessarily always have to be. In fact, that's what's going to be the second part of the lecture. But so the dimensionality in typical is high, right? I'm standing here, I've got a bunch of joints, I'm moving around, there's a huge number of motions one can make. I've learned that most people don't understand the reference because I'm old enough, but Monty Python Ministry of Silly Walks. This okay, good. A physics audience, I can always count on a good laugh. Good. Excellent. I feel slightly less old now. And then basically you can have this wide variety of motions and movements that you can possibly make, and this is a high dimensional thing, but maybe when you're actually modeling it, it could be lower dimensional. And so the other point you made was about variability. And of course, there's a lot of variability in these other things that we've been mentioning. In fact, that's basically all of a lot of the stochastic processes work is actually measuring, like, how well can I measure this gradient based off of the fact that there's random molecules hitting me. But there's something fundamental that there's variability across populations, across individuals, and even within the same individual, like within the same task. So if you measure experiments with a lot of behavior, you see, in general, you tend to see more variability than in other types of experiments. What else? Ilya?
You honestly don't even know what to measure. I mean, like, what is the thing that we're measuring, right?
Well, so that right. So that's the question, right? So, but why can't we do that?
I mean,
That's more what
I
I'm.
know what the genes are, I know what the spikes are, I know that they sort of matter, but here I look at the roof and it also has a hair moving around, and presumably this doesn't quite matter, but if I take movies from there, right, I don't know what's relevant.
Right. That's not all variables relevant. Right. Anything else? Yeah.
So there are a lot of timescales.
Yes, exactly. Yeah. So. Many time, and I'll also add and length scales. Right. So in particular, we have a lot of these things. Right. There's a lot of time and a lot of length scales. And I would add my own personal one, and this isn't something that you would probably experience having sort of grown up in your nice cloistered physics environments, is basically anthropomorphism, which basically a fancy word for pretending these things are like us. Right? So we want to put a lot of our own, and it's a really strong tendency, even after doing this for a decade, right? I still find myself doing this every once in a while, of wanting to put your own sort of intuition and feelings behind what you're seeing, because these are pretty charismatic animals a lot of the time. And this has a tendency a lot of the time to make people jump towards more qualitative answers. So this is less sort of technical, but a more sort of sociological reason, but important nonetheless. Any other thoughts? Yeah. Yeah. So I'm going to put it this way. There's sort of, to build off of this, there are very long time scales, right, including memory. So animals can be moving around, and there's something that, like you have this Proustian thing of some Madeleine that you ate as a child, and then you smell the thing, and it all comes rushing back, right? So and then this force, and then this sort of intrinsically causes you to go out and get another Madeleine. So right, yeah.
Environment is complex.
Yeah. So right. Exactly. I mean, this is there's a lot of external feedbacks, and just in general, you don't always know what the sensory information is. So to make even if it wasn't complex, even if you just have some light stimulus coming in, unless you designed your experiment very, very carefully, you don't even necessarily know what that stimulus is. Yeah?
They can learn and adjust the behavior. Like they can learn and adjust their behavior.
Yeah, adaptability. Right. And in some ways, that goes into variability. Because it's not like you have some nice quantum system, assuming there's no like hysteresis or measurement or anything weird like that. You can take the same thing, measurement a bunch of, well, quantum's a bad example. Take some nice, take some, I am at a condensed matter somewhere, collateral. Take some nice, like an ideal gas or something. You look at it a bunch of times. The measurement should always be the same, or it should always at least be drawn from the same distribution. You can't always count on that. There's a lot of non-stationarity when you're talking about behavior, and what I'm going to argue throughout these 4 lectures is that it's inherently dynamical, and that a lot of we've built up a lot of statistical infrastructure over a lot of years, and there's this tendency to rush into physics and use all of that nice sort of physics statistical information we have. And that always isn't necessarily the right technique. And really moving towards this more dynamical picture in some ways is a way of dealing with these bunch of length and time scales and other sorts of things. Any other thoughts? All right. This is basically all I had. In fact, even a couple extra ones. This is great. Okay. So, and I would to get towards Ilya's point, and maybe I would put this in a slightly different way, is there's no language for behavior. I'll phrase it as a question just to make ourselves feel better. What is the language for behavior? Right. And what I mean by that is analogies. And despite the fact that I talked all about anthropomorphism and these other issues over here, one of the key interesting ideas, and one of the things we're going to try and do when we're thinking about behavior, is to try and actually think about behavior in terms of what a useful analogy is and how to build in infrastructure that we've created from all these other fields into our understanding of how these animals are moving around. Okay. Now, for a case of analogy right now, not for analogy for behavior, but analogy for what behavior lacks, let's say we're going to look at DNA.
Sorry, can I?
Yeah.
Before we go in that, right? Do we agree? Maybe it's even a question for everybody. Do we even agree that behavior, on the definition of what behavior is?
I'm about ready to get there. Right? Yeah.
So ethologists have been studying behavior for a really,
Yeah.
really long time.
Not as long as you'd think, but yes. So only since like the 1950s, really. Yeah, Tinbergen and Lorentz were, well, maybe 47, I think, was the first really ethology paper.
Okay. But people have been studying animal behavior in some shape or form for some time.
Yeah, and we're going to, yeah.
Okay. Now I was just asking, like, what is it that you as a physicist or quantum biologists are trying to do that is different from what physicists have done?
That's a great question. Hold on to it. Because that's going to be sort of in a few minutes. We're going to talk about that, right? But that's exactly sort of what I'm going to try and get at. And mainly what we're going to do is get at this through example, because there aren't that many examples. And we're going to work with one particularly good example, I think, today of showing what you can do, thinking like a physicist about these problems. And then the next lecture, we'll see what happens if you think poorly like a physicist throughout those problems, and that'll be my work. So, all right, great. Any other questions? All right. So think about this. So if we're talking about DNA is a remarkably complex object. Right? There's whole lots of people that still today study pulling on DNA and measuring all of its physical properties, and understanding exactly what DNA is a really complicated thing. However, if you have a bunch of geneticists in a room, or physicists that are talking to geneticists, and they say, okay, you should measure something about that DNA, what are the things you would measure? Sorry, I'm hearing some things. Base pairs is one example, right? Right, you'd measure GA, T, or C, or you'd measure, say, equivalently, you could translate it through the various codons and get amino acid sequences instead. Or if you're really super fancy, You could measure things like a chip-seq, where you're measuring basically like transcription factor bindings. Right. But there's these sets of numbers, right? Do the base pairs tell you everything that's happening with that DNA molecule? No, very, very far from it. Right? Because some of that stuff open to be transcribed, some of it's closed, right? You saw Leonid's talks. Okay. So the idea is DNA is this really complicated object, but you go and measure it, and you know sort of what are the first things you would want to measure. Okay? I go out and I measure those things, and the point isn't that these end the argument, is that these things start it. You have some things you can measure it. You can then measure, okay, between this set of organisms, that set of organisms, you see this distribution of base pair mutations or these deletions or crossover linkage and things like this. And you can then go and sort of build theories and models. And this is a very nice quantitative language to describe what's going on. Right? And it's a very low dimensional one. Right? DNA is highly, highly dimensional, but this is very low. Now, similarly for neurons, neurons, again, just by construction, because they have DNA in them, are even more complicated. Right? What are some of the things that people in neuroscience would measure about neurons that would have the similar sort of flavor? Spikes, right. Right, so spikes, or even, so even more, so slightly more complicated than spikes. Yeah, membrane voltage, right. Of which spikes are a discretization of that voltage. Or you could even go further and say like firing rate, where you average a bunch of spikes together and figure out roughly how many spikes you're going within some certain interval. Right. So again, this doesn't tell us everything. Right? As I'm sure Stephanie told you about, like there's a lot of stuff in neuroscience that just even if you were to measure the spiking from every neuron in the brain, no one really knows what to do with that. Right? But even then, you sort of know that there are these low-dimensional things, and you can learn quite a lot from them, and you know those are things to measure. Behavior, right? There's not really, I mean, this set of low-dimensional things. Like one example of a low-dimensional measurement you could make is what we talked about earlier, and you could do things like measure, say, center of mass kinematics, right? That is a low-dimensional measurement, but it loses a lot of information. And so really what we want to do, in particular when we're thinking about these, is we want to have how do we take this complicated creature and then reduce it to, represent it in a manner where we can really make theoretical and experimental interrogation on sort of the level that we as physicists like to do, and where our theories we can truly take seriously. And it's not just, oh, that thing sort of increases, that thing sort of decreases, but no, I want to put a bunch of points and fit a line through it, and that line is theory. That's not a fit, right? This is sort of what we want to do and what we're aiming at. Now, are we there? No. But the point is sort of how do we even set up the problem? And really that's what these lectures are going to be about. It's less about the solution and more about how we even start to set up the problem in the first place, because that's really where the field is right now. All right. So that being said, and so now to get back to your question over there, right, of course people have been studying behavior for a very long time. Right? I'm not going to be standing here, well, I am sort of standing here as the arrogant physicist, but I'm not going to be claiming so much to be standing here as the arrogant physicist to say, oh, these guys all sucked and they didn't do anything before. No, like there's been a lot of really fascinating, interesting work. And if you think about how it's been measured typically, there's sort of three traditional ways that it's been done. And this goes back not just to the ethologists, but even a little bit further when you talk about people from experimental psychology and behaviorism and other sorts of fields. Right? So the first thing you probably see the most, if you were to pick up a random paper from some journal where they're measuring some behavior, what you'd probably see is something like a paradigm. Okay? So what do I mean by that? I mean this. Like standard, you have a mouse in a maze, right? It's in a maze. It can turn left. It can turn right. I have encoded the behavior I want to measure into the experiment itself, right? I'm just measuring whether it turned left, whether it turned right. I could have measured also like how long it takes to make a decision before going to left and going right, or other things like that. I could have made a monkey go and have a task where it has to hold its arm in the middle, and then move out to one sort of region. But I've designed sort of what the task goal is in my measurement by sort of this experimental paradigm that's been created. This has probably been the most common and the most widely used form of measurement in sort of behavior, in field of behavior, both in behavioral neuroscience, behavioral genetics, and also in psychology as well. So this is certainly one form. The other form gets more along the lines of the center of mass movements, and we'll call them coarse measurements. So here's a few examples. So these are a bunch of examples here from fruit flies. And what I mean by coarse measurements are things where you've taken it upon yourself, without looking at the data, to reduce its dimensionality. So we're going to say, all right, let's measure the center of mass of the fly, or we can measure its velocity. So these are a little small, but you can measure, for example, on the most coarse level, this is survival on the y-axis, so how many died, right? That's a pretty coarse measurement, and sometimes it's the useful measurement to make, right? Or you could measure like number of times they successfully completed something, like how many times they're able to climb up the side of a wall, or how many times they crossed something in the middle. This is very common in circadian rhythm experiments where you see animals, you're trying to measure how active they are, and you basically see, you put a little laser beam in the middle, you just count how many times it crosses the laser. Or my personal favorite is this thing here. This is to study, believe it or not, because people do this, alcoholism in flies. And this device is called the inebriometer. So this is going to be chemistry, actually. So what you're going to do is you put a bunch of flies in this baffle, and basically what they do, and so the flies have to be actively paying attention to stay on the baffle. And what happens, you're going to flow various amounts of alcohol in there, and at certain points they pass out, and they're going to fall out. And so you can measure the mean elution time of the flies out of the cylinder. So I mean, and of course, like this in some sense this is ridiculous, and I think they're sort of owning it by calling it the inebriometer, but at the same time, right? It's a way of saying, did the thing pass out or not? Right? In a sense, is what it's measuring. Right? Which I mean, and there are obviously advantages to this. Right? And just like in what we talked about with the paradigmatic approach, right? You can get really high throughput. Right? You don't have to put a huge amount of work in. Like it takes a lot of work to design the experiment and to train the animals. But once you've trained the animals and done all that other stuff, then you can just take data for a long time and it's repeatable. You can tell your friend how you did it, and they can hopefully do it the same way. And so this at least allows for some degree of repeatability and a large amount of data. Right. So the third way is really, if you go towards most of the ethology literature, so what was alluded to is ethology, and so ethology is basically the study of well, it's always a hard thing to define, but I always like defining it as the study of behavior within its natural context. So as opposed to studying how just an animal can push a lever, it's about actually understanding what are the evolutionary and neural and genetic bases and environmental bases of how animals behave. And this was some famous work that was started by Konrad Lorenz and Nico Tinbergen and Karl von Frisch, who won the Nobel Prize for this in 1972 or 73, and which apparently, according to was like the most surprising Nobel Prize that ever was awarded, because no one was ever expecting these random field biologists to win something for medicine. I'm still surprised, and I wasn't even born. So the idea is, so we have this third method, which is what I'll call intuitive, and this is really what people have been using in ethology. And what this is sort of the grad student's nightmare. What you're basically doing is you have an animal, you have a trained grad student or postdoc, you stare at that animal for a really long time and say, the animal does 7 things. Not 8, not 6, 7. And basically what you can then do is then you go and you sit and you watch you've written down, you codified what those 7 things are very precisely. as you can see here. See this mouse grooming. We have shimmy, rapid vibration of body, large synchronous but asymmetric strokes of forelimb over top of head, momentary interruption of active movement with forelimbs at chest height, and then they say a more detailed description of these components is available. Right. So, again, it's repeatability. So I don't really mean to make fun, but it just is to hammer home the point that you've sort of created this infrastructure, and then how it used to be is you would go and sit down, and every time that maybe you hire undergrads to do this, maybe the grad students don't have to do this, but every time the animal does something, you put a little check mark box down, and then you make these things called ethograms, which are it's doing this behavior, then that behavior, then this behavior, then that behavior again, and so on and so forth as a function of time. So you can do a little bit better than that now. So now there's all sorts of fancy supervised machine learning techniques where you basically now you just need to sit down and at the computer for a little while and press a button whenever the animal is doing the behavior you're expecting it to do. And now you can train basically a classifier to say, okay, this was backward walking. Right? Backward walking. And the computer goes on, does this, and now you can do this in super high throughput using billions and billions and billions of images, which is great. But even still, and this is in their own abstract, the users encode their own intuition about behavior by annotating. Again, given the lack of any language, I don't know what else you can do, honestly. Right? It sort of sounds funny when I'm talking to this, and I know I'm sort of preaching to the choir to a bunch of physicists, but at the same time, this is a difficult problem. Right? Thinking about how do you actually go and watch these animals doing really complicated things. Ever watch a mouse groom? There's all sorts of crazy things happening at all sorts of time scales and all sorts of length scales. It'll move around, and then the decision of what to do next and what to do next after that is actually more complicated than what you would imagine. And so they had to just rely on this intuitive method when doing this. And this has been really the state of the field until maybe about 10 years ago when people started to think about these types of problems a little bit more seriously. So that's sort of how we're going to try and think about this. And how we're going to try and think about this is to actually look at the data. Right. How can we actually look at a data set and from that data set say something about what an animal is doing as a function of time and how we can describe its movements? And to do this, I have a very odd task because I'm not going to, it's not so weird that I have to describe somebody's work that's not my own. This is general par for the course for teaching. The problem is I have to basically describe the work of somebody else that's sitting here in this room. So we're going to talk about Greg's work. So we're going to talk about the worm as an example. The worm C. elegans. I can never pronounce. How do you pronounce the genus name? Canarhabditis. Right. Right. Yeah, C. Yeah. They always just go by C. So C. elegans, and basically these are these soil-living nematodes. They go and they swim around. We'll see a movie of them in a second. Right? And maybe that will sort of give a sense of why we're working on these. And I should also note that most of what I'm going to be talking, there'll be the two papers, Stevens et al., 2008, and this is PLOS Comp Bio, and also we'll talk again, Stevens et al., 2011, and this is PNAS. And if we have time, I'm also going to talk about a different paper which builds off of these called by Andre Brown and colleagues from 2013, also in PNAS. I am wearing down this chalk quickly. All right. So let's actually look at this. So what the experiments are, and so these were experiments that were done by Roux, who at the time was at Princeton, and he was basically studying this little guy. So this is a C. elegans, crawling around on a plate, has sort of this traveling wave which moves down the body, and you put it on this agar dish with a copper ring around it so that it sort of stays, because they don't like copper very much for some reason. Do you know why?
It's poisonous.
Ah, that would be a very good reason. Again, not my work. So it's a very good reason. And so then basically what you're doing is you're tracking, and Will is one of the world's experts at making a tracking microscope, and he's done this for bacteria. So you can actually watch and move and see as this sort of eye goes around here. But what we're going to study is primarily what's going on this side. So we're going to try and find a measurement for what that worm is doing as it crawls around. And what we really want to do is we want to uncover sort of, and I'm going to use this term incredibly pretentiously, say what are the essential dynamics of it? Like, how can we describe or the basis of how it's moving and how it's interacting, and how it's moving through its environment as it goes? So we have this worm. Go over here. Actually, sorry, Greg, I'm going to erase your citations already. So what we're going to basically do, I'll turn this off so it's not a worm running around. All right. And at a given time in a field of view, what we're going to have is we're going to have a worm. And I am an absolutely atrocious artist, so I apologize in advance. Catherine's back there. She has to deal with me drawing in group meetings. So. I should have just made a slide, but I'm too proud. All right. Let's say that's our worm. Right? That's not exactly a very worm-like shape, but let's say it is. Now, the question is, how do we actually, what we want to do is we want to measure this worm's posture. So by that I mean not just where it is. Let's say we don't care about where it is. Let's make a choice one. We want translational and rotational invariance. Basically meaning that if I'm going to call one behavior this over here, if I'm over here, I want to call it the same thing. So we're going to make that choice. And really, a lot of this, like all of these other things that Michael was talking about in his lecture yesterday, is about making choices. How do you decide what aspects of this thing to represent and what aspects of this animal to throw away? And that's really going to be the big theme over both today and especially tomorrow. Okay. So choice one is we're going to talk, we want to define behavior in a way which is independent of position, right? And this isn't necessarily great. Like, you are probably more likely, and it's more salient to see turning behavior when you're near the copper poisonous thing at the edge. But we're going to ignore that. In reality, whenever I say we, I really mean Greg. But but it's somehow grammatically easier just to say we, because it's us. We're talking, and so we're going to have to make this choice about how we're actually going to represent this thing. So as we represent this thing, right, what are some of the options? So one, right, we could say, what's the center of mass? What's an obvious problem with this other than just this lack of description? Worm people are allowed to answer, Leo. You're smiling. What? There's lots of problems, right? A particular one. Yeah, that's what I was looking for, right? The center of mass in this guy is probably like here, right? So it's not even in the worm. Now, you could fix that a little bit by doing that and saying, okay, instead of talking about the center of mass, you could talk about the sort of, I don't know, center of center line.
Why is it a problem that the center of mass is not in the
It's somehow aesthetically displeasing. It depends on what scale, right? If you care about where it actually is within the environment, maybe it's not a problem. But if you're trying to actually measure something about what the worm is doing, and maybe it could be like if the worm is curved, then the center of mass is like completely outside the thing by a fairly large margin. It's not actually describing anything about where any part of the worm actually is.
But I'm just wondering, is
Right.
that informative in and of itself, or no?
It could be, right? It tells you that there's a more interesting dynamic than the center of mass, right? Yeah.
Why is what is the center of center line here in that aspect?
It at least makes it the point is on the worm.
But then, like in every case that you mentioned, knowing that the point is inside doesn't actually help.
It might. It's hard to say, right? We're not going to do either of these things. Right. I'm just trying to build up possibilities, right? Because really what I'm trying to emphasize is the choices. The choices are being made, because this is going to be a recurring theme about how choices get made and sort of how I want to approach the worm and how I'm approaching this work as an example where those choices are made a little bit more straightforward because of aspects about the animal. So there's 3, which is instead of doing this, you could just measure the whole center line. All right. I'm running out of the big chalk, so let's go to the small one. All right. So you can measure the whole center line, right? So we now have a 1D curve parameterizing the object. Or the fourth one is you could just measure the whole area, right? Right. This really is some 2D thing filled in. Right. This is a 2D structure. And you could actually just measure, you could parameterize this not as a line, but as some complicated 2D shape. And so what Greg and his colleagues decided to do was to focus at this scale. And this sort of seems like a sensible thing to do for a couple of reasons. One is that for the most part, the animal is well, if you watch it move around for, yeah, do you have a question?
You could also technically just segment the image and look at the distance matrix, right?
Right. You could take the image and represent it as a distance matrix. This is as a distance matrix between pixels in the image. You mean? Yeah. Of course. Yeah. You could also. Right. I'll sort of toss that in within area. It's sort of a. Well, maybe we'll say 5 topology, right? Because if we look at the distribution of those distances, that uniquely specifies a particular topology or shape, right? Right. Yeah. You could have done that as well. What?
That loses no information.
But it's not unique necessarily, right?
Yeah, it's translationally and rotationally.
It is translationally, rotationally invariant, right? It potentially loses no information. But I guess the point I'm trying to make, and what the choice that they made, was they actually wanted to lose some information, right? Right? And that's what a lot of us do. That's what a lot of things we do in physics, is we throw stuff away. Sometimes we could do it more principally than others. Like if you look at the renormalization group that Bill talked about on Friday, right, in RG, that's explicitly a whole mathematical formalism for deciding what we can throw away. Right? So in some sense, we're deprived of that here. But we're going to try and actually move away from that and define things in a way where we can throw away information and still see how well we can do. Okay. So what Greg and his colleagues did is they decided to look at the center line. And there were 2. I can't speak for exactly what the motivations were, but I can say what the 2 in my head would have been. One is, and probably the most obvious, is the fact that you don't see too much variability sort of along this way. Most of the motion you can well describe as a center line movement. And that's sort of, if you actually look at the musculature of the animal, that's sort of how it's designed, to sort of have these center line modulations, and so it's a reasonable approximation to do on a first pass. The other thing which is useful is that this is so the Reynolds number of this thing. So for how many of you here know what a Reynolds number is? Oh, good. I can't always tell about fluid mechanics with a physics audience. So the idea of a, maybe that was covered by a previous lecturer. Okay, good, excellent. Who covered fluid mechanics? Oh, Mimi?
Yeah.
Okay, great. Okay, great. So the idea is you're at this relatively low Reynolds number. And what that means is that your movements don't have a history dependence. And so that means if you make a small movement along a particular direction, you don't have to worry about the overall history of the kinematics. You can just worry about what that movement is right now. And there's a whole line of theory called resistive force theory that people have thought throughout the years. And this works very well with the idea of looking at a thin line moving through something. I don't know if that was part of the motivation, but I think that's minimally is another reason why this approach was likely to be successful. Again, why I'm taking time on this is, and I'm just going to keep saying this, is that these choices matter. And as you're looking, particularly in systems where they're as complicated as an animal moving around, at every point you're making choices. And then you go and present it later at a conference and make it look like it was all magic. But in reality, this all a set of very difficult choices that you're always having to make. So we've decided to look at the center line. So now we have, in some x and y coordinate, we have a worm that sort of looks like this. So now we need to make a decision about how are we going to parameterize that line? We have this line that has a beginning and has an end. And so let's say this is the head, and this is the head, and this is the tail at the other end. Right? And yes, these worms do have a specific head and tail. They have most of their neurons up at one end. So this is the head up here, and this is the tail back at this end. And so how they chose to parameterize this is by, you can break up this guy into a bunch of different segments. In fact, they broke it up into 100 different segments. So you have 100 segments. And then at each segment, what you're going to do is you're going to draw a line and measure the angle, basically the tangent angle of that with respect to the ground. Right? So you have a bunch of different, so this isn't a great drawing because it's mostly really terrible angles to draw. Again, sorry. We have a bunch of these different thetas. And so you have this set theta 1, theta 2, all the way up to theta 100. I'd like for all of you in your evaluations for next year, for this, to put colored chalk at the chalkboard. I can sort of work with, oh, is there? Oh, there is, excellent. All right. So, apologies, Leo. And so what you can see is, right, you have a bunch of these things. And we've measured these with respect to the lab frame x-axis here. So what's the one problem we have with this representation? Sorry? Too many. Well, that is a problem, right? So problem one, there's too many numbers, but we're going to fix that problem. So the question, if you couldn't hear, was why not just do a Fourier decomposition? So part of it is because this is a finite size. So you have a finite size curve. A Fourier transform is going to look terrible. Right, because you're going to basically get ringing effects on the edge, because you basically more or less have one wave on the body, maybe a little bit more. And so you're going to basically, if you parameterize it with a Fourier transform, and I don't know, did you try this at some point? Yeah. I imagine you're probably a huge amount of ringing doing that way. Yeah. And so it's probably not particularly clean, and also in some ways it's going to be a little bit less interpretable because you've tossed away some, because now you're in a complex variable space instead of a real variable space. What's the other problem? Having to do with our invariances. Which invariance does this violate? rotational, right? Because this whole thing, I could rotate this by some other angle, and it's just the same. And so basically all we're going to do is say that at each time point I, we're going to basically subtract off some number just to make sure that this equals 0. And that's just to satisfy that this thing is rotationally invariant. So at each time point, what we're going to do is we're going to calculate all of these angles, then subtract off the mean so that it has a mean of 0. I'm not going to keep subtracting off the mean because it just adds a bunch of terms to the stuff we're going to talk about. But just remember that this is always happening. But now, as Vasil was saying, right, the other problem still exists. This is 100 numbers. You look at this thing. It doesn't feel like it should take 100 numbers to describe the posture of this animal. So we got a bunch of numbers. We're a bunch of physicists here, and what do physicists like doing with numbers that are on a matrix? We diagonalize them. Yeah. Well, you could. I mean, essentially, this is sort of we're measuring it as a continuous curve, right? You could interpolate it. Right? But the point is, and there's other things one could have done. This is probably the easier, right? Because you could have made it a continuous curve and then do some sort of functional data analysis. But why deal with complicated functional data analysis if you don't have to? Right? A lot of this is making your life easier, right? As there's always, this isn't the sort of thing, like part of it is maybe an attitude coming into these types of problems is you're not going to find an exact solution, right? You're not going to be able to go down and write an exact solution for the brain. Right? Well, some people might think you can, but even for a worm brain, you're probably not going to be able to go and write down the whole thing. And as my colleague over here has a nice quote on his door about a map maker making a map which is exactly real space, which is exactly in the same scale as real life. So at that point, it's not a particularly useful map. Right? So the point is to try and actually, can we get rid of stuff? What can we toss away as we go? And how can we make our lives easier as physicists? And my attitude, and one of the things that I like so much about this paper and why this paper was inspirational to me when it came out was thinking about, I like too dumb to fail approaches. And by dumb, I don't mean stupid. I mean relatively straightforward, right? Things where you didn't have to add too many epicycles of creativity around. Can you just take the data, do the simplest possible thing to it, and see what comes up? And that's I think, really what the attitude is going to be here. Tomorrow, you're going to see that when you get to an animal more complicated than a worm, the simplest thing you can think of is still really a pain. but that'll be tomorrow. That's your fun for tomorrow. But, right. So we have this. Right. We have 100 variables. And we're a bunch of physicists here. Let's make a matrix and diagonalize it. And the matrix we're going to diagonalize is the covariance matrix. Right, so this meant matrix Cij. Right. Just your standard covariance matrix of looking at the relative variance between two angles I and j. So we have a covariance matrix, and then what we can do is actually see, is there a lower dimensional structure in that? And when we look at a covariance matrix and diagonalize it, what's sort of the popular term for this? PCA, principal components analysis. Right. So what we're going to do is diagonalize this. And that's basically PCA, and we're going to see if a low-dimensional structure pops up. And because, again, I can't draw, I've just copied the figure. And what you can see is the following. So here's the covariance matrix over here on the left. And you can tell immediately looking at it, that is if you've looked at enough covariance matrices, like some of us have, this is very low-dimensional. If this was high dimensional, if this was 100 dimensional, how would this look? Yeah, it would look like sort of like speckled noise, right? But this has some really nice, clear, sort of banded structure that banded structure already tells us that there's going to be some sort of like periodic boundary conditions moving along as we see this. It might be difficult to see from the back, but there's a band here, there's a band here, there's a big band in the middle. And so we can sort of see this, and sure enough, what we can measure is we can measure the eigenvalue spectrum of this matrix. Again, it's a positive, definite, symmetric matrix. Or it's symmetric matrix, therefore it's positive, definite. So you've got a bunch of non-zero eigenvalues, or greater positive, semi-definite eigenvalues. And what you can see is that this is the fraction of variance explained. So how you interpret your eigenvalues in PCA is each eigenvalue is the variance you can explain along that eigenvector direction. So you can see the first one is maybe about like 0.3 or something, 0.35. The second one you get up to like 0.6, 0.7, and then third, fourth. And by the time you get up to the fourth, you've explained 95% of the overall variation with only 4 numbers. So now we don't have that many numbers. We only have 4. We've gone down from 100 to 4, and 4 seems a lot more plausible. And moreover, we can then look, so what's represented here are the eigenvectors, more or less, transformed back into a nice frame. But you can sort of see that what you're looking at is the first, second, third, and fourth eigenvector. The first and second one almost look like a sine and cosine pair. Right? They're not quite sines and cosines, but they look really close. And they're sort of off-shifted by a little bit. So let's keep that in mind. And then the third one looks like it's a body bend. And the fourth one has a little bit more complicated structure. Yeah, Ilya.
So I usually complain all the time when people sort of say, oh, it looks like something, right?
Mm-hmm. Well, we're going to make it more precise in a second.
Okay.
Right. I agree. I'm pointing it out as inspiration. So I agree one shouldn't present your work as this looks like something, but this looks like something can be a way of making a choice. Right? It's a way of thinking about what decisions you're going to make to reduce the data. And I think that looking like a sine and a cosine made the authors of this, and I don't know if this was planned in advance. Again, I'm trying of inventing a history here because I don't actually know what it is. But
I love listening to this history. It's perfect.
I know. Well, because the thing is, I'm going to give an honest history tomorrow of my work.
It's all good.
And so what you can see is you can look at those two, and it almost looks like a sine and cosine pair. So the next thing might then be, can we actually see what would happen? So let's pretend for a moment, again, throwing stuff away. Let's ignore the third and fourth modes for now. Let's ignore we're going to ignore modes 3 and 4. Okay. So if we do that, let's actually think about these. So we have vectors, and we have our eigenvectors, and we have these eigenvalues. So let's actually look at the first two. Let's look at the projections of the data. All right, so each data point we can now represent, because all these eigenvectors are a rotation of our data set. We can now look at the eigenvectors, or each data point is a projection on these first two eigenvalues, or these first two eigenvectors, rather. So if we look at these projections, so our projection we'll call AI of t. And this is just simply going to be, this is our vector of 100 thetas that we've measured after doing our mean subtraction, minus the mean minus the mean value for each of the thetas in particular, and then we're going to dot that by our eigenvector of choice. So here we'll just do I equals 1 and 2. So what we can then do is say, let's look in the A1, A2 plane. And let's say that these are exactly sine and cosine. What's our data going to look like in this plane? Sorry? Sorry. So if we have just A1 and A2, right? And we think that's all that's really happening. And those two things look like a sine and the first two modes look like a sine and cosine relation to each other. What's going to actually wind up? What is it going to look like? What? A circle. Yeah. We're going to sort of expect the data to kind of look like that. And as a matter of fact, it does. So this is just a 2D histogram of the A1 and A2. So A1 down here, A2 up along the vertical. And you can see there's this very nice histogram of where the data actually lie. Now note there's some interesting fading over here and here. That could be due to a couple of reasons. One is it could just be it's slow here, then moves fast, right? We didn't say that they actually have to be moving at a uniform thing around this. Or it could just be that there's also really the third and the higher moment, sort of it's rotating out of the plane, so you don't exactly see it here. But that being said, you don't see much here, you don't see much here. Everything is largely around that circle. This tells us that we do sort of have the sine-cosine relationship. So let's make our lives even simpler. Now what we're going to do, going from 100 dimensions, we went down to 4, down to 2, and now we're just going to go down to 1. We're going to measure a phase variable phi, which is just going to be equal to the arctan of A2 over A1. So we're going to measure this phase variable. Now, we've described the whole dynamics of this worm all the way down to a one-dimensional variable. That's pretty cool. In and of itself, like that in itself, you probably can stop and be happy. But let's not. You can think about understanding these dynamics, and there probably were other ways you could have arrived at the same thing. But the point is, this was, I think, a good systematic way of getting there. You looked at the video. You made a choice of how to represent the animal in that video, and then you followed that representation down to sort of its logical conclusion. Here. So now we're going to talk, instead of talking about the whole complication of a worm, it's 302 neurons and all the stuff going on, we're just going to talk about phi of t. And ideally, what we'd like to do is to model this. Can we have a model for how phi of t is going to evolve with time? So one of the nice things is you've already seen a Langevin equation, and so we're going to do exactly like that. We're going to try and write down. We're going to we'll call it a Langevin-like equation for phi. And we're going to assume in that form, right? So if we're going to write it down in this form, we're going to say d phi dt is just equal to some angular velocity omega. And we're going to write down from this the second order differential equation, stochastic differential equation, d omega dt. And d omega dt is going to have two parts. So part one is going to be some function of phi of t and omega of t. And part two is going to be another function of phi of t and omega of t, but now multiplied by some noise term eta of t. And we're going to assume eta of t looks sort of white noise-ish. And so we'll have that it's going to look something like that. Or at least that's the goal for how we're going to try and derive this. And the question is, can we actually derive what this function is? So we have our So this is our deterministic part. And this is our stochastic part. we're going to try and derive these from data. And so, yeah. Well, it is additive, but it's just because this is changing as a function of position, right? So it's basically you're changing the noise amplitude as a function of location, right? Right. So it still is this white noise process that you're adding to this deterministic part, but it's but you're going to allow for the fact that that noise could be a function of phi and omega as well. All right. Great. Any other questions? So in practice, what we're going to do is we're going to measure phi of t, and then you can do some filtering, and you can thus also measure omega of t. I'm not going to get into how you could actually filter it. There's a gazillion ways one could do this. There's fraught choices in that, but that's whole another story for another day. All right. But let's say we can measure phi of t, and we can measure omega of t reasonably well. And given that, what we want to do is we're going to try and fit these parameters. So one of the things we're going to assume is if we assume so if we're going to assume that this equals 0 for all t, and for all phi and omega, basically that our noise term has a mean of 0, then that means that our root mean square, thus that we can say that our root mean square maximum likelihood estimate of f of phi and omega is just equal to this conditional omega dot given phi and omega. Right. So you're basically going to take for each imagine you had an infinite amount of data. You could take your data, put it into a bunch of infinitesimally small bins, average the value of omega dot, and that's going to be your average for f. Okay? Now in practice you can't really do that, because A, you don't have an infinite amount of data, and B, that data is somewhat noisy. And so so what they did, and there's a bunch of ways one could do this, but how they decided to do this is to fit it to a functional form that was sufficiently broad. So they fit to a form f of phi of omega equals m So basically you have a constant, a polynomial term, and then a Fourier series term. Right, and then you basically take that f is the real part of this. Right. And this is a sufficiently general form because you have a polynomial for your dependence on omega, and then you're also going to have this phase dependence, and this sort of makes sense. You would want to have some periodic function for the phase dependence because it's a phase. So, and they got these numbers 5, minus 5 to 5, and 0 to 5, I believe through cross-validation. Is that right? So again, Greg and David Schwab agree that we should cross-validation is a good way of doing these sorts of things. And again, there's other types of functional forms one could have done, but this is a sufficiently general one that you're not going to sort of box yourself into any particular corners. You've just preserved this phase symmetry by looking at by having this Fourier-like term. And then the polynomials essentially are allowing your data to look like as much as a fifth degree polynomial for each of the different things. And then given that you have that, I usually hate writing this low on this one raises. Excellent. I'll lower it again in a second. But so then given that we have this, our noise term is then just equal to what's left over So basically this squared error term between the measured omega dot and then f of phi and omega, which you measured up here, and then conditioned on phi and omega. So here you are actually grouping your data up into bins and finding what the average variance is around that. Okay. Any questions about this? So if we actually do that, so these are the functions you get. So here's your f and here's your sigma squared. So on the left over here you have your f of phi and omega. This would have been a little bit clearer if you would have used a different color map, Greg. But that's fine. So, but what we're going to look at is, let's actually look at this. Let's ignore the stochastic part for a minute and just think about the deterministic part of the system. Right? This is just a second order ODE. So you have a second order ODE. Oh, what's the first thing you always do when you have an ODE? Look for the fixed points. Right? That's always the first thing you do. At least when there's one in front of me. I very rarely have the hubris to actually figure out I can solve it. So let's actually look at what the fixed points are. So first of all, vertically, where do the fixed points on this plot have to be? Sorry? Here, right? Because the fixed points you have to have, this is omega and this is phi. And so because phi is defined as d omega, or as omega is defined as d phi dt, thus you can only be a fixed point along this line where omega equals 0. So we can look along this line, and now we need to see where the zeros are. And so it looks like, well, 0 is this greenish color. So it looks like there's a 0 here, a 0 here, and a 0 here. Now that you have fixed points, now that you have a fixed points of a differential equation, what's always the thing you do with the fixed points? Are they stable? Right. So is this one stable? So we can sort of see, well, up here, if I go down, it increases. So that would push me back upwards. Right? So that means it sort of looks stable this direction. If I go up, it gets bluer. So that means it would decrease. And so this one looks like it's stable. This one's the opposite, right? If I go up, it goes up, and so that's probably unstable. If I go down, it goes down. Yeah, that's definitely unstable. And over here, this also looks stable again, right? As I go up, it goes down, and so that's going to push me back down. If it goes down, it gets higher, and that's going to push me back up. And so it looks like we have a stable fixed point here, a stable fixed point here, and an unstable fixed point here. Moreover, you can sort of see probably towards the edge, this is partially probably the limit of fitting what happens when you fit a polynomial to these types of things. You're going to get some amount of fluctuations over here, which I don't necessarily know if they're real or not towards the very edge. But you find the worm very rarely is out there anyway. Moreover, over here, if you look at the noise, you can largely see that there's more noise sort of down in here and a little bit less up there towards the top. And that actually might be instructive, right? So, of what's actually going on. So let's actually look to see what trajectories happen in the system. And so what you can see is, so this is just a bunch of trajectories that Greg ran through this. And you can see, sure enough, there we have a stable fixed point. We have a stable fixed point. Notice how the eigenvalues look a little bit different. But nonetheless, you still wind up getting a couple of stable fixed points. And you have this unstable, sort of homoclinic-like looking point towards the middle, which sort of wraps around. And up top, you have this separatrix, where you have motions up here. And bottom, you have another separatrix, where you have motion down here. So let's look at this top red curve. What is that? What's the worm doing on this top curve up here? Yeah? No. So here's 0 omega, right? So we're going to see that this is so this is positive, right? So we're seeing that it's a positive angular velocity, meaning that it's moving forward. And it never drops below 0. So you can sort of see what's going to happen. You're going to start point here, and then the worm is going to keep on moving forward. So this is going to be forward locomotion. Similarly, this is the other. right? So now here, omega is negative, and it never goes up above. So this is going to be backwards locomotion, right? Because you're always going to go from here, because your angular velocity is negative, you're going to move back in this direction.
I missed why omega, like omega is the speed of phi, right? The derivative of phi, and you're also calling it backward and forward motion. So is phi like the position?
So phi means that you're basically having a traveling wave moving on the body, right? Because you have the sine and this cosine. Right? Maybe I didn't explain that clearly enough. Right? So phi is basically means you have advancing means you have the sine wave, which is traveling, and that sort of means that the animal is moving forward. And then, but that sine wave can also go in the other, or that sinusoid can also go in the other direction, and that's moving backwards. You can see it's not a perfect line, but this is also data, right? So it's a model fit directly to data. So it's pretty impressive. And then these two things are nothing, right? These are basically pause states. Because here, the angular velocity is 0, and it's at a particular phase. So at this phase, and these two things, these are places where the worm isn't moving at all. So you have forward, backward, and then two different ways to not move. There's actually some interesting consequences to this, I think, but I'm not going to really mention those too much today. Right, and there's been some other papers which have also independently, completely different types of models of worm behavior, which have also come up with these two different types of states, and there's some arguments from the neurobiology as well. But interesting. And the other thing on this plot, just to mention, is one of the things you might want to characterize to make sure that your models actually fit reasonably well is that you want to look at the autocorrelation function of what's left. Right? So what do you actually measure for your noise, this eta of t from your model? And ideally, this should look sort of delta function-ish. It's never going to look exactly like a delta function because, again, it's data. But you can see that originally your autocorrelation function for omega of t has this relatively long time scale, whereas for eta, it drops pretty precipitously and rings out. So it's about as good as delta function-ish as you can rationally expect from data. So you actually have this approximation of reasonably separating out these timescales is actually a decent one. Okay, so now what? Right. So this is cool. We have basically from just direct measurements, we've set up the stochastic differential equation. What can we predict from it? And that's what I want to spend the last, I guess, 11 minutes talking about. There's a few different predictions one can make. So the first prediction we're going to talk about is reversal frequency. Okay. So by reversal, what I mean is how often do you go from the forward to the backward state? And how this looks in the data is it looks something like this. So if you measure phi as a function of time, you'll see it go up. Sorry. Lecture's been going on for a while already. All right, phi goes up, then it goes down for a little bit, then it might go up again, and maybe down, and so on. So you get these sort of distinct points where you see reversals. And what we'd like to know is to predict something about so if tau is the amount of time that it spends in this forward state, tau 1, tau 2, and so on, can we have a probability distribution of those taus? Does this model actually reproduce that well from the data? And so we're going to make, again, some assumptions. And so we have this stochastic and this deterministic part. We can see these are the forward trajectories up here. But every once in a while, we can have enough noise to push over the fixed points and move in over here. And so the question is, how often would what's the distribution of timing we would expect to stay in this forward locomotion state from the model? And we're going to make some assumption. And the key assumption that Greg made is that we're going to say that noise is small compared to the barrier. So basically that these transitions are relatively rare. Right? They're relatively rare so what this buys us is we're essentially assuming that those flipping between the forward and the backward state are more or less independent of each other. That each time it does it, it's its own independent event, and it's independent of the history of the past reversal that's made. That's the assumption that goes in. So this is a room full of people that have taken a lot of StatMech courses. So given those assumptions, what sort of distribution would we expect that to look like? Chemical reactions? I can hear a lot of whispering in the back. Sorry? Arrhenius, yeah, exactly. We'd expect this to look like, we'd expect P of tau to look something like e to the minus tau over the mean of tau. Right, we expect it to sort of to have that type of form. So we can actually measure that from the data. And from the data, what you can actually see, and I'll just put the plot up. Oops. Skip that. All right. So from the data, what you can actually see, so this is the actual data over here, and you can see that it actually fits. This is on a semi-log plot. And actually see there's a little bit of a curve, but for the most part, it fits a line pretty well. So that looks like an exponential decay, and the mean tau from the data, they're picking about, they measure about 16.3 seconds, plus or minus some. So now the question, what exactly would the model predict? And what they did was something a little bit clever, is they assumed that essentially as you have this term, you have this term over here, what you're going to do is multiply this by some beta. So you're going to essentially say, more or less, as you increase or decrease the noise, what's going to wind up happening to this rate of switching that you would measure from the model. And lo and behold, you also get this very nice exponential decay with some weird exceptions up here at the front. And that's just because when you get so low, is basically you can never hit the barrier, I think. So you just stay in the forward state for way longer than you would naively expect. But for the most part, you get this line, and not only that, you predict the slope shockingly well. I'm surprised why is the error bar here so big, Greg? Right. So the point is that they're still close, right? Well, to a physicist, they're both 16, right? Right. So they're both very, very close to each other, and so that means this model will actually predict something which is non-trivial. And not only that, which is really cool, is we never put in this model that you had to have reversals. Right? The fact that you get these events like this is a function of you see this in the model, and you didn't actually ask the model to do this. All you asked the model to do was to fit these variables as best as possible. Okay, so that's one. The second thing, if we go back.
Gordon, sorry. Can you comment a bit on you tried to model the system at a dynamical system, right?
Mm-hmm.
And then it seems to be the range conditions are fundamental probability, right?
Well, but it's a stochastic differential equation, though. Right? There's a stochastic term, right? Did that answer it? Okay. All right. All right. So the one thing I really want to get at in the last 5 minutes is something which is going to be key for tomorrow. And the other thing that you see from this is the concept of stereotypy. And so this is basically behaviors that you see often and repeatably. And looking at this, looking at this map, where in phi would you expect most of your transitions to occur between the forward and the backward state? Sorry? Yeah, somewhere around here, right? Because you don't have to cross this whole crazy fixed point. And so the idea is maybe this dynamical system is actually predicting something about the stereotypy of the behavior as you move between one and the other. And so essentially what you can do is you can say you let t equals 0 be when the reversal occurs, and then you can basically look to see what's happening to that distribution. What's the distribution of omegas and phis exactly at that time and then at previous times towards that? And what you see in the model is, and this is that probability distribution. So here you have time and you have phi on this axis. And you can see that, sure enough, at t equals 0, it's not quite actually at phi equals 0. It's a little bit below it, but it's reasonably close. And you can see that almost all of the transitions sort of take this category here, move down, and so on. So you have this very stereotyped behavior that almost every time it's making the transition, it has a structure that looks like that. And if you look at the data, it's a little bit blurrier because, again, data. But you can see essentially the same structure, whereas basically blue bands there, and then you have these heights in between. And so the idea is you have the stereotype behavior. And really, as we get into tomorrow, stereotype behaviors are going to be the key bit. And what I'm going to try and convince is going to be the key component for how we can have a language describe behavior on a more sort of general scale than just in the worm. And I'll add one more thing, since I have a couple more minutes, is that you can also say, well, let's actually see what's happening if I fit this dynamical system at different times. Because the worm's getting hungry, right? What you're doing is you're putting the worm on this plate. There's no food there. After a while, it's going to start changing its behavior. What's been noted a lot of times previously is at short times, it'll sort of just hang around, sort of like bacterial chemotaxis, run and tumble stuff. Short time scales when it's not hungry, it'll sort of hang around locally in a dish. But as it gets hungrier and hungrier, it'll start zipping around and doing a lot more straight lines. So that's going to mean it's going to reverse less. And sure enough, you can take the first third, the middle third, or the last third of the data set and fit different models to each of them. And more or less, this f function doesn't change. So the deterministic part is roughly the same. But what you can see is that actually the noise part decreases with time. So what this means, so early, middle, late, is that noise part is decreasing, meaning that you're seeing fewer reversals. It's harder for the worm to flip back and forth across those sort of separatrices in the middle. And that, in fact, is one way that you might predict if you're looking inside of the worm's physiology, is you might then look for ways for that randomness might be generated in locomotion. Yeah? If you would decrease the total amount of what? The food. Well, no, the food is just, there's no food. This is, you put it on a dish and it starts getting hungry. You take a very happy, well-fed worm, you put it on a dish, and after a few minutes it says, hey, you know what, I'm hungry. I need to start finding food again. No, there's not like a chemostat or something where you're actually changing it. This is just, it's getting hungry. Right. Okay, so there's, of course, a bunch of other stuff you can do. And then what we'll talk about next time is how do you try and extend this idea towards when you have an animal which has a more complicated morphology than a worm. And we're going to move after that into thinking about how do you go from these sequences into building these dynamical structures. So, thanks.